Welcome back to the deep dive. Today, I want to start with a quick visualization exercise.
Okay, I'm ready.
So I want you to just look at your fingertip and imagine right on the ridge of your fingerprint a tiny cube of tissue, about the size of a grain of sand.
1 millimeter on each side. So tiny, if you dropped it, it's gone forever.
Exactly. But here's the thing. That single grain of sand, which is a piece of human cerebral cortex, contains 1.4 petabytes of data.
It's a number that's almost impossible to wrap your head around. It's like 300 million high-res photos in a speck you can barely see.
It's completely staggering. And this is the HO1 dataset, a collaboration between Harvard and Google, and it's our mission today to dive into it, because this is supposedly the highest resolution map of the human brain ever.
And it really is. I mean, we've had brain maps before, but they're like looking at a country from space. You see the major highways.
Right.
This is Google Street View. You can see every house, every mailbox, every single power line connecting them.
And when you zoom in that close, some of the textbook stuff starts to look a little shaky. We're going to talk about neurons that act like compasses and connections so powerful they seem deliberate.
It takes the idea of brain complexity from an abstract concept and makes it, well, something you can actually count and measure, which is a huge leap.
So let's start with where this sample even came from, because that, to me, was one of the first surprising things. This isn't from a brain bank.
No, and it couldn't be. The issue with cadaver tissue is that it starts degrading the moment the heart stops.
And you can't map connections at the nanoscale if the structure is falling apart.
Exactly. You need living tissue. And in this case, it came from a 45-year-old woman who was undergoing surgery for epilepsy.
Okay, so how does that work?
Well, to get to the part of the brain that's causing the seizures, surgeons sometimes have to remove a small, healthy piece of cortex that's in the way.
Like a plug of tissue.
Precisely. And the research team was right there in the operating room. The second it was removed, it was put into preservatives. The cells were essentially still alive when they were fixed in place.
Which explains the insane level of detail. But then there's the engineering challenge. I mean, slicing a 1 millimeter cube into more than 5,000 sections.
Yeah, and each slice is about 30 nanometers thick.
What's a good comparison for that.
A strand of DNA is like 2.5 nanometers. So we're talking about slices just 10 or 12 times thicker than a DNA molecule. It's just absurdly thin.
And then they had to take a picture of every single one, which I read took 326 days.
Almost a full year.
A year.
And that wasn't with just some standard microscope. They built a custom rig with 61 electron beams scanning at the same time.
So even with 61 scanners running 24/7, it still took that long.
It's just a monumental amount of data to capture. Can you imagine the stress? If the power goes out on day 300, you might lose the whole thing.
I can't even think about it. But getting the images, that's only half the battle, right? Now you have 5,000 2D pictures.
And you have to put them back together. You have to trace every single wire from one slice to the next. A neuron's axon can look like a tiny dot in one image.
And a slightly different dot in the next. And you have to know it's the same wire.
Doing that by hand for 50,000 cells would be impossible. It would take centuries.
So AI comes in.
Right. They use something called flood-filling networks. The AI looks at a cell in one slice and then predicts its shape in the next slice and sort of floods it with a unique color to trace it through the whole volume.
It's like reassembling a shredded document that's in 3D and has billions of letters.
That's a great way to put it.
So once they did all that, they could finally take a census of the neighborhood. And this is where my old biology textbook really got thrown out the window.
Ah, the old neuron-centric view of the brain.
Yeah, neurons are the stars. Galilea are just the boring support staff. But that's not what they found at all, is it?
Not even close. In the sample, the support staff outnumbers the stars 2-to-1.
2-to-1.
For every one neuron, there are 2 glial cells. And the single most common cell in the entire cube wasn't a neuron at all. It was the oligodendrocyte.
The cells that make myelin, the insulation.
Exactly. There were over 20,000 of them.
So what does that tell us? Why so much insulation in this high-level processing area?
It tells you that raw computation isn't the only thing that matters. The brain is obsessed with speed and timing.
It's not just about sending the signal.
It's about making sure it gets there at the exact right moment. Myelin speeds up the signal. Having this much of it means the brain is meticulously managing the synchronization of everything. It's a high-speed, heavily insulated circuit.
And speaking of infrastructure, the plumbing in this thing. The blood vessels.
Oh, the energy grid, yeah.
They found 230 millimeters of blood vessels, that's almost 9 inches, coiled up inside a grain of sand.
It's a bit of a paradox, you know. The brain runs on, 20 watts of power? It's incredibly efficient overall.
Right, dimmer than a light bulb.
But locally, the infrastructure to deliver that power, the glucose and oxygen, is just massive. It shows you how metabolically expensive thinking is. The brain is always just moments away from an energy crisis.
Okay, so it's a high-maintenance, high-speed environment. But what about the design, the architecture? This is where we get to the compass cells.
Yeah, this is where it gets really interesting. Going in layer 6, the deepest part of the cortex, they looked at these triangular neurons.
And they're dendrites, the antennas for receiving signals. They weren't just pointing every which way.
No, not at all. You'd expect some randomness, like tree roots spreading out, but these were incredibly organized. They showed a bimodal distribution.
Meaning they pointed in two main directions.
Two specific mirror image directions, like a compass that could only point north or south. And they were perfectly aligned with the big white matter fiber tracks running underneath them.
So why? Why would the brain care which way a dendrite is pointing?
It's about managing information flow. Think of it like this. You have data streams running from the front of the brain to the back and vice versa. If your antennas are random, you pick up all the crosstalk. It's just noise.
But if you align them,
You can selectively tune in to a specific direction of traffic. It turns the deep cortex into something less like a tangled mess and more like a printed circuit board with dedicated lanes for information.
so there's a real rigid geometry down there that we just never saw before.
We just never had the resolution to see it this clearly.
Oh, okay. So the cells are aligned, but then there are the connections themselves. And this is the part that I think really stopped me in my tracks. The super connections.
The outliers, yeah.
My understanding has always been that a synapse is a whisper. One neuron connects to another. It's a tiny vote, a small influence.
And that's true for the vast majority. In the data, 96% of all the connections were just a single synapse, a whisper.
But then they found the screamers.
They did. They found pairs of neurons connected by not one, not two, but in one case, 53 separate synapses.
53. I mean, my first thought is, isn't that incredibly inefficient? Why not just make one really big, strong connection?
It is metabolically expensive, for sure, but it's all about reliability. A single synapse is probabilistic. It can fail. It's noisy.
So if you absolutely, positively need neuron B to fire when neuron A fires,
You can't rely on a whisper. You need a detonator. You build in redundancy. This connection cannot be allowed to fail.
but how do we know this isn't just a fluke? Maybe the two neurons just happened to run next to each other for a long way and kept bumping into one another.
A great question, and they tested for that. They ran a null model, a computer simulation where they kept the neuron shapes but randomized their locations to see what would happen by chance.
Did the random brain make any super connections?
Not a single one. The probability of a 50 synapse connection happening by chance was calculated to be less than 1 in 10 billion.
P-value of less than 10 to the minus 10. So this is not random. This is design.
It's biological intent, and you can see it in the structure. The axon doesn't just run parallel to the dendrite. It actively wraps around it, like a vine climbing a trellis. It sends out little branches to grab on again and again. It is physically hugging its target.
That image of a vine, it feels like something that was built over time with purpose.
And that's where this gets really profound. It leads to this idea of engramics.
The search for the enneagram, right? The physical trace of a memory.
Exactly. For a century, we've had this theory that learning physically changes the brain's wiring, but we've never been able to point to a specific connection and say, there, that's the memory of your first pet.
And you're suggesting these super connections might be exactly that.
It's a very strong possibility. Think about it. When you practice a skill over and over, you strengthen that circuit. Maybe it starts as a whisper, but through years of repetition, the brain builds that vine, adds more and more connection points, until it's an unbreakable detonator-style bond.
So we could be looking at the physical embodiment of a deeply ingrained habit or memory from this patient's life.
We could be looking at the physical scar of learning. It's an incredible thought.
It really is, but we also have to talk about the other strange things they found, the literal oddities. They had a whole section on UCOs.
Unidentified cortical objects, which is just a fantastic name.
It is. They found things that are not in the textbooks,
Yeah.
like axons that just curled up into these massive tangled yarn balls.
Whorls, yeah. Huge knots of wire leading nowhere, and strange egg-shaped bodies inside cells that no one can identify.
And what about the broken bridges in the blood vessels?
Right. A capillary that just stops. A dead end.
So the elephant in the room is Is it just what an epileptic brain looks like? Are we just seeing pathology?
That's the billion-dollar question, and the researchers are very clear that this specific chunk of tissue appeared healthy under a normal microscope. It wasn't the seizure focus.
But the brain as a whole was not healthy.
Exactly. So maybe these whorls are scar tissue from the electrical storms of seizures. Or a side effect of medication. That's one possibility.
And the other possibility?
The other, more unsettling possibility is that they're in all of our brains.
That this is just the normal messiness of biology.
Right. We have these clean, idealized diagrams, but nature is messy. When you zoom in this far, you're going to find glitches, debris, construction errors. We just don't have a baseline from a perfectly healthy human at this resolution to know what's normal chaos and what's disease.
It's like we've landed on a new planet, and we can't tell which plants are native and which are invasive weeds.
That's a perfect analogy. We have no field guide yet.
Which brings me to the thought I really want to end on. We spent this entire time talking about 1 cubic millimeter.
What?
One.
And a whole human brain is about 1,000,000 times bigger than that.
1,000,000 times. So if we found compass cells and detonator synapses and mystery objects in this one single grain of sand, what on earth is waiting for us in the rest of the beach?
Well, that's the zettabyte problem, isn't it? To map a whole brain at this resolution would generate more data than currently exists on every hard drive on the planet combined. We couldn't even store it.
So we're actually limited more by our computers than our microscopes at this point.
For now, yeah. But this HRO1 dataset is the proof of concept. It shows what's there to be found. And for me, it just changes how I think about my own mind.
How so?
I can't think of it as a computer anymore. Computers are neat and tidy. This is a jungle. It's a galaxy. It's teeming with strange structures and superhighways and cosmic dust. It's just so much wilder and richer than we ever gave it credit for.
A galaxy in a grain of sand.
Exactly.
I think that is the perfect place to leave it. We will, of course, put a link to the NeuroGlancer viewer in the show notes. You can actually fly through this data set yourself online.
Oh, you have to. It's a real trip.
Thanks for joining us for this deep dive. And next time you have a thought, just try to imagine the millions of gigabytes of machinery that just spun up to make it happen. We'll see you next time.
Take care, everyone.